So last but certainly not least, today we've got stepping in because Dr. Robert Horn unfortunately couldn't make it for us this afternoon, but stepping in at the last minute and it's still from the same department, in fact, working with Robert, I believe, and you're going to present some joint work and some of the same vision and things. We've got Dr. Viktorija Makarvaite here talking to us around kind of some things around biosensors and kind of sustainable sensors and looking at the future of that and kind of how her work fits into the wider picture and how we're innovating for this future here at Kent. So without further ado, over to you. And while you're setting up, I will just say, please, as always, put your questions down in either the Q&A box at the bottom or in the chat, and I will pick them up and I'll ask them in a conversation at the end. So,
right, let me just share my screen as I'm starting. So I am Dr. Viktorija Makarvaite, and I am one of the lecturers in biomedical engineering. So a lot of my work is sensors for medical use. So for the hospital, as well, a lot of wearable technology. So I do, a lot of infection control, a lot of device development for basically infection control. So I will talk to you about specifically some passive biosensors and kind of where we're taking them to make them a bit more green in the hospital setting, as well as where the technology itself is moving in the future and how the Kent University is basically taking it there. So let's kind of get started. So a little bit of an overview. So this will be, I will kind of talk about this as a two-pronged approach to medical devices. In a way to reduce medical waste products, waste of medical products. I'll incorporate this with talks about implantable tags for passive RFID-based communications and sensing. When we talk passive, that just means it's battery-free. So there is no battery involved in any of my devices that I will mention. And I will kind of mention back and forth each time about the benefits to clinicians. So a lot of it is a price, it's cheaper. You can do long-term monitoring while the devices are in the body. It's easy application. They usually tend to be when it's passive. You don't have to have all these extra components often. So they tend to be less cumbersome, a bit more flat. And I also will mention future projects as well. I'll show you a little video at the end of how we're printing stuff now and actually making a patient-per-patient case. So there's a lot of interest going on when it comes to these types of projects. And the University of Kent is actually really well known for wearables. So Professor John Batchelor, one of my supervisors, my previous project, now I do a lot of projects with him, as well as Dr. Robert Horn, who was supposed to do this original talk. We work a lot together, and Professor Batchelor himself specifically is known, and he's the leading expert on passive technology and wireless sensors for medical applications and this type of sensors. So, we really have a really strong team when it comes to this type of research. So the examples I'll give you is a joint collaboration between the East Kent Hospital, the Kent Cancer Trust, the Kent Fungal Group at the university. That's something unique that we have at the university. We have a fungal group, as well as the part where I'm from is the Communications Research Group, and there's obviously subgroups between that. And Robert Horn and myself are part of the antennas group that goes under the Communications Group. As well as a lot of the funding, like I said, it comes from the Kent Cancer Trust as well as the EPSRC funding. So, there's a lot of a lot of people involved in getting these projects out, especially when it comes to any kind of medical applications. And as a biomedical engineer myself, the one advantage that I have is that I have the medical background and I also have the engineering background. So for example, my PhD was electronic engineering, whereas my actual my bachelor's and my other two masters were all medicine. So I am actually licensed in medical technology to work as basically a clinical engineer in this country. So I have a lot of medical as well as engineering background. So this allows me to kind of almost be a go-between the biosciences as well as the engineering. So yes, apply for biomed. So what is the project that I'll be talking to you about? So the first project is really the voice prosthesis project that both I did, and it was part of the Chem Fungal group. And a voice prosthesis is the little device that you can kind of see here. When a patient has their larynx removed, you need to put a little device inside in order to allow the patient to both breathe and to be able to talk. Sometimes you maybe have seen movies where they have or know anybody who has like a little voice box to help them basically talk. You can also talk just by putting your finger over the hole, and it allows sound, basically vibrations to occur and sound to come out of your mouth, basically to actually be able to produce speech. The issue with these devices is that they can become colonized. And if they become colonized, they stop working. So I will kind of go through a little bit of all of this just a little bit. Let me show you a quick video. This is one of the main companies that actually produce these voice prostheses. And this is just a quick video of how they insert it.
Before you start the insertion, be sure.
We don't need to need the sound, but I'll just show you the video quickly, because I think it's a very interesting understanding. So obviously, before you can insert it, you have to have surgery in order to remove the larynx. So that's a very invasive. The actual changing of the device is not that complicated. It's usually done either by nurses or speech therapists. But obviously, if it gets infected, you have to change these devices often, and that leads to increased cost. There is some studies going on to figure out, why some people get infected, have their voice prosthesis colonized much more readily than others. It actually has been linked to alcohol use as well as eating a lot of acidic foods. So there's a lot of different links that can cause your prosthetic device to be colonized way more quickly than you would normally need to. But so it's a really interesting little video showing you how you actually insert the device. And obviously, if you close up the hole, you can actually talk. You snip it off so you can stays in there. Obviously, if it gets colonized, what happens is there's like a little flap inside that moves air back and forth. And let me just go to the next slide so you can see it. So there's this little flap in here this is what allows the air to go back and forth. And when you have colonization, so this side is what sits towards the back of your neck. So this is what actually, when you eat and drink, comes in contact with this surface. And when it becomes colonized, this little flap that allows the air to go back and forth can no longer move. So the device becomes basically useless. So you no longer can talk, your breathing is affected. It basically becomes useless. So there have to be ways to try to find how can we improve this, basically. How can we reduce colonization? How can we reduce basically, having to throw away these devices? Because once it gets to this state, you have to go back, you have to have it changed. Depending on how bad the infection is, sometimes you need revision surgery. So you have to literally go back, open up the neck, remove debris, so basically any kind of necrotic tissue due to the colonization, and have to have a much larger voice prosthesis actually put in place, which leads to more issues, more spillage, and there's a lot of issues that occur once you have to have revision surgery. So and also it's just really costly to the NHS. So if we can reduce this, it's very helpful, obviously. So there were two prongs approach that we tackled this problem. So the first approach, we actually studied and found ways to improve treatment. So this is actually published by one of the PhD students, Daniel Petland. He looked at different antifungal activities and medications, and he wanted to see okay, so how, if you use this drug or this pathway, or you use this combination of drugs, how can you actually reduce colonization and kill off the actual species so that you can extend the lifespan of the voice prosthesis? So this was the first approach, trying to figure out what kind of drugs can we actually use with the patient to reduce colonization, to extend the lifespan. This was published in 2020, so this is just past year that we actually finally published it. But this research, as well as with working with surgeons and speech therapists at the hospital, has led to basically us increasing the lifespan of these original devices by 270%. So that's a really big improvement. Some patients were having actually to have these voice prostheses replaced every like 6 to 8 days, some were 2 weeks. Most people were under 6 months, and we were been able to extend it to at least 6 months for a majority of patients. So it's a big improvement and a big reduction in waste of these voice prostheses, because we don't have to spend money on them if the patient still has them. And a lot of these voice prostheses can go as high, just for the prosthesis on its own, is about 100 quid. So if you can imagine, every couple of days, you have to have this patient come back to the hospital, see a speech therapist, have it replaced again, and then go back to, it not only does it cost, it also, stops the patients from just having a normal life. If they have to keep going back to the hospital, going back and going back, it's not good for the patient either. So we were able to increase the lifespan of these devices. And because of this research, we actually developed a pathway that is used in clinical, so in all hospitals. This is available for all speech therapists, as well as physicians, to use to like actually improve treatment. So we tell them, if you have this type of colonization, use this drug, or if you have this, use this drug. And we've actually been able to improve healthcare, not just in the UK, not just in Kent, but all over the world. So we have these being utilized in Australia, in Nigeria, and Scotland, and like all over the world. They're actually using our pathway that came from this original research. So here's just a little example. You can find this, like I said, here. This is all available online. This pathway is available for you to have a read. Feel free. And I'm just showing an example. And here's just an example of a patient whose life has really been improved by this specific pathway and this kind of research. He used to go every 2 weeks to have this voice prosthesis changed. And now he hasn't had a change, I think, in 3 months or so. So this, it's actually helping patients. So everything I told you is amazing. It's already in clinical use. The drug therapy and everything that we developed is already in use. However, what is the issue with just step one? So this is just the first prong. Like, what is the issue with it? Well, the issue is that we must have the voice prosthesis fail before we can implement the pathway, because that pathway is for people who can have continuous failure. So they have to keep failing a couple of times before the clinician starts using the drugs that we recommend, because it's actually drugs directly for the prosthesis. So because obviously if you don't want, we don't want to increase antimicrobial resistance, so we don't give it to all the patients. It's not just automatic. The patient has to have failure first. And obviously, if you have to only do this here, you can never catch the pathway early. So you can never catch the colonization and the infection early. You have to wait till it fails, till the patient goes in, either has surgery or has the prosthesis removed. So this adds additional costs. So we thought, is there a way to monitor colonization live? Can we have a patient that we put it in the device, something on the device, and be able to actually monitor the colonization as it occurs? So we thought, maybe there is some kind of app we can make going with a sensor in the device to kind of go towards more of like a smart hospital setting or a smart home setting. Because there's a lot of interest in this health sector of going into the Internet of Things, as well as monitoring patients live and, to basically improve patient outcomes. So we were really interested in, okay, what can we do? We know that we have the treatment, but how do we give the patient the treatment when they need it, so we don't have to throw away the voice prosthesis once it's fully colonized and no longer functional? So we decided to look at, into live monitoring. And I personally work with UHF, so ultra high frequency technology. And I do a lot of passive sensors. So we thought, okay, maybe we can make a passive sensor that can detect infection, and we can be monitoring it live. So first, the first step was basically trying to figure out, okay, so how does colonization, because the main species that forms biofilms, specifically on voice prosthesis, because they're present in your normal oral flora, and these are the ones that cause for the device to fail whenever we remove them, because part of the original study was getting actual patient samples and testing them to see what's there. We saw that the main species that forms these biofilms that destroy these prosthetics was Canada albicans. So I needed to really figure out, how does this grow? So we saw that within 0 to 6 hours, just based on literature, based on our own research, we saw that it kind of attaches first. 6 to 8 hours, it starts to almost like form this what's called a germ tube. So this is basically, it's elongating. So you can see that it's starting to form a germ tube. And about 20 hours, it keeps, colonizing and colonizing the surface and colonizing the surface. And it starts producing these, it has a little bit of water channels in between. Water channels just means the media can go through towards the surface. And then you're colonizing and colonizing till basically the whole surface is fully covered around 20 hours, which is where I consider intermediate growth. And then it keeps forming these hyphae. So those germ tubes keep elongating. And then they have this extracellular matrix form, and there's some sugar, some DNA in between. It's just basically like this, almost like a sticky mess. It's just really thick layer of biofilm that forms. Okay. So basically there's early, we consider that in 3 phases. So there's early, there's intermediate, and there's mature growth. So that's how I was like, okay, can I detect early? Can I detect intermediate? And can I detect mature? So the first way was, okay, so you can kind of see a live view of that, what that occurs, see how it grows and it extends really heavily. So we thought, okay, how do we determine, so we know how it grows, but how do we determine the electrical properties in this, when that grows? If I made a device, how would it be able to actually read that growth? So we utilized what's called an Excelligence RTCA machine. So this is, you can buy it off the market. It's very expensive, but it basically uses these wells. So you just put the cells in and you kind of watch them grow. So I made like a biofilm attachment process, and I watched them actively grow onto the sensor just to see what happens to the electrical properties as it starts covering up and start going through basically the early to intermediate to mature. So I just wanted to see what happens. And we got this really amazing, that very distinct growth curve. And it's a bell growth curve. So basically, as it starts increasing, we kind of see it early to intermediate. It goes up. Cell index here is just the way the system is read. It correlates with impedance of the system and capacitance. So I'll describe what we saw. But basically, we saw this really unique bell curve as C. albicans biofilm forms. So as it goes into intermediate, it kind of levels off and then it goes down. And we saw that obviously this growth occurs up to here once it fully covers the surface. Once it fully covers the surface, we see this drop. And it's very unique to just C. albicans. I haven't seen, we tested Staph aureus, we tested E. coli, we tested other human cells to see what happens. And most of them usually, once they grow, they kind of level off and stay level. Whereas C. albicans does this bell curve appearance. And we wanted to see why. Why does this do that? I just wanted to add in that, sorry, this is a bit out of order, but it doesn't matter. I'll talk. That for the voice prosthesis, the reason why we can't just continue doing this is because obviously this is not live. This is not in the person. This is growing on a device. And a normal prosthetic for the voice prosthesis, the activity, the colonization actually occurs here. So we can't really, use that for live monitoring of anything. That was just for us to figure out what's occurring in the system. So basically, after way more studies and using some different types of sensors that I've developed, and you can buy off the shelf as well, we saw that as the biofilm thickens, there's a clear law in what is called capacitance. So that was the electrical properties that we saw of the biofilm. And once there's a full coverage, it almost like decouples itself, and there's what's called a shift occurs, and you see what's called a second resonance. All that is saying is that we can detect it. So I know what it looks like. I know how it grows. I know what's happening in the early, intermediate, and the mature stages, which means if I know this now, I can develop a sensor for it. Okay. And like I said, because this is a distinct bell curve, it means any kind of biofilm that has this bell curve, I can very easily detect. So before I started developing, there were some issues that you have to consider when you're working about any kind of embedded system, not just passive, but in general. You have to consider the electrical properties, the dielectric properties of whatever tissues are going to be around the prosthetic. So that's a big issue in any kind of embedded. That's why it's so hard, and there's not many people in this field who are experts. So that's because there's a lot of work involved with this. So you have to either develop multiple devices that can be applicable to all different people, because the electric properties change depending on usually say somebody who has more fat versus more muscle. So every single person can have different dielectric properties. And not only that, every single location in your body can have different dielectric properties. So something that you put in your arm might not be able to be put in your stomach or in your leg or anywhere else in your body. So you have to take this into consider, like where are you putting the device and how is it going to function? And the easiest way to obviously overcome if you have an embedded wireless system is to just put a battery. A battery gives you more power. You have more power. The device can function. The signal can more easily basically come out. Obviously, I can't do that because I work in passive systems. So it's not possible for me to add a battery. So I had to really develop a sensor that was robust enough to be able to function in multiple people, or develop multiple sensors that can work in multiple people, basically. So this complicates the use, of passive design. So you kind of, this thing you always have to keep in the back of your mind: where are you putting? What type of tissue is surrounding it? How close is it to the skin? Because the closer it is to the skin, the more the easier it is to send out a signal, basically. And so like I said, you need to consider that not only the type of microbial growth. So you have to first figure out, okay, so what are you trying to grow? What are you trying to detect? But you also, like I said, you have to consider where you're putting it. A very interesting thing is that I got lucky. I'm just going to say that. Turns out that when we tested the necks, because all my devices are directed out of the hole, this area of the neck, between different variety of people doesn't change all that much. So I tested volunteers with BMIs from 20 to 44, and all of them had a very small changes in the dielectric part where the voice prosthesis actually is. So I literally could just design one device and it would function. Obviously, this would not be applicable anywhere else in the body, but we have this speak probe that we can test the dielectrics of different like phantoms and body tissues and stuff. So I saw that people had not much variability when it came to the neck. So when I was designing my sensors, I made sure that the sensor was directed, the wireless, the reading was directed directly to out of the patient, so that I didn't really concentrate. Basically, I try to reduce the side lobes. If you're more if you're more technically minded, I try to reduce the side lobes so that everything is directed out. So all my directivity is basically going out the hole and just directly towards the reader. So that helped me. And obviously, I'll give you right now from the paper, the average like permittivity conductivity of the patients barely varied. So I got very lucky with working with the neck. And how do we implement all of this knowledge? So now I know how microbes grow, how the biofilms grows. I know that the neck is a perfect place for doing a single device, a prototype. I know that I need to direct everything out of the neck, like straight out. So how did I actually incorporate all this knowledge and make a device? So I implemented this, and this is a paper that's already published, so you're welcome to read it. I'll go through a little bit of it and what we found. And basically, I'll show you some graphs. It's a bit complicated. Here, it's just showing you that within the frequencies that we work in Europe, as well as Europe and China, it has a minimum of about 665 centimeters out of the body. So I can read it about 65 centimeters outside of the body. That might not sound like a lot, but all the previous publications have it a limit of about 20 centimeters. So the fact that we were able to utilize the hole to increase the read range is really important. So I don't want to undersell this. This is very very big in this type of study, basically. And this is the device. So the original research obviously was done with what we call a phantom. So the phantom is just usually like a gelatin mold to mimic a neck. And this is the device. You can kind of see the device, and you can see how it was placed together. So I had the tag over the prosthesis. Then I had a bit of a polymer layer to protect the tag. And then we had basically an artificial biofilm and then saliva on top. So this is how it was tested, basically. So like I said, this is directly from the paper. So you're welcome to, go back and read this, just to kind of find out a little bit more. And it basically functions like this. So you have the sensor on the device. Biofilm growth starts occurring. It starts occurring on the prosthesis. You get infection, and what happens is that it alters, what's called the system impedance, which is also we're looking specifically at capacitance. And by having this kind of change in capacitance, we're able to detect infection. So that's all going back to the original research. So this is how the sensor basically functions. And this allowed us to basically detect this kind of Canada albicans biofilm formation as early as 8 hours equivalent. So obviously this is all tested, in a prosthetic device, in a phantom, but it would be equivalent to about as early as 8 hours of infection. And also this works pretty much in any UHF frequency range. So it could be Europe, it could be US, and it could be Japanese. So it could be adjusted to work pretty much in any frequency that you would need in the UHF, obviously. You can move on to Wi-Fi or Bluetooth or anything like that with a specific device. The issue with this is that if you look at the paper, is that obviously it's the first prototype, so it's not as user-friendly as I would like. So it still relies on read range and frequency shifts to detect it, in order to determine what the capacitance is. So I'm like, okay, this can't be very usable for clinicians. So there's a new prototype that I'm working that is actually smaller than this one. I can't show it to you, sadly, because of the publication rules. But it'll be smaller than a 10 pence coin. It has a steady read range because it has its own embedded capacitance shift that it could occur. That just means it can adjust and read when there's a shift in capacitance rather than needing a shift in frequency. So it's a bit more stable, and because you have this output of capacitance, it can be correlated to a code that can be correlated to like an app or a program that would automatically give either the patient or a clinician that readout showing that there's infection or it's still healthy. So everything here, obviously is prototypes. It's still in the works. We don't have a clinical study yet, but we are working that. So hopefully, we can reduce the voice prosthesis device waste by recognizing early infection. That's the overall goal. So that patients can be put on treatment pathway to extend the lifespan of the device. And we're hoping to extend the device lifespan past the 6 months that we currently have. We're kind of limited because we use this drug called Makarvaite, and it's a gel and it has alcohol in it. It's a very small amount. You're not going to get drunk off of it or anything. But because the devices are polymers, sometimes it could cause a bit of expand the polymer slightly. And it could cause deterioration that way. So that's why we have it limited at 6 months. However, if you detect it early, we might not have to go to Makarvaite. So we can actually extend the pathway, so you can get stuck at the first step of the pathway rather than at the bottom end of the pathway. So you can actually extend the lifespan way past 6 months, as long as you don't have to go into our end-all drug, which is the Makarvaite. And obviously, the next step is also to finish up that new prototype. Obviously, COVID has made it a bit more difficult to actually do all the testing. And the next step after that will be a clinical study. So, this is where we're currently at right now with this type of research. But on another project that is kind of correlated with this is that what if you could just put the medication into the prosthesis itself? So we're working with Dr. Simon Holder from the material sciences, from the CISC group, to actually develop a polymer that can not only kill the colonization when it attaches, but actually either kill it or stop it from becoming infectious. So you not only have treatment for when it is colonized, you have a device that detects it, and then you have a polymer that actually kills it before it becomes infectious. So it's like now going into a three-prong. But all of this is collaborations. How are we going to work together to make basically patient care so much more? And here we have this polymer that we made that actually either stops or kills 87% of all growth. So here is what a normal standard polymer looks like when you have Canada growth on it. So this is actual pictures that we've taken. And a new polymer, you can see here that on the standard, it has these germ tubes. And when you have filamentation, it means it's becoming infectious. And the one thing that I haven't mentioned is when they become infectious, when they have this, they can actually grow into the polymer. So not just clump up on it, they can actually grow into it and damage the polymer that way. Whereas our material causes basically less than 30% of them to be allowed to filament. And the ones that usually over time, they are also killed. So once they're there, we don't allow them to become infectious, and we actually kill them by just changing the way the polymer is made. So it's all about side groups. It's the side chains of the siloxane polymer, basically. So this is where a lot of that research is also going. And let's finish up with talking about a bit of the future. So I'm going to show a bit of a printed technology. So right now, a lot of the work that I'm doing is looking at basically made-to-order sensors. So how do you can print directly per patient, per clinician request? So let me see if I can share my screen and let's see. Where is it? Give me a second. I'm just going to stop sharing for a bit and open up from here. Give me a second. Just need to find it. There we go. Okay. Where is my Google? There we go. So this is something so this is a video from printing that we did yesterday, just to kind of show you the idea. I sped it up. So hopefully everybody can see. Yes? Yeah. Okay. So this is a machine, I think it's called a Ventura, that can print directly any kind of, use a silver ink to basically print the design that you need. So we can actually make per request per patient. So this is just an example of one. We're just showing you less complicated printing. And then we're going to show you a bit more complicated device printing. So let me just go. So this is what we're currently working on next in order to actually improve and make our technology a bit more green, because obviously if you don't need to have a giant manufacturer involved, and if you can have different, maybe smaller manufacturers just making per request per patient, you can not only save money this way, but you can also basically go a bit more green, because you don't have to manufacture a bunch of these if they're not needed. You can actually adjust them per patient. You can maybe change the material. So for example, if somebody's allergic to some kind of side group of like nickel or anything else that you need to worry about, you can change the inks. You can change the material that it's printed on. You can change, you can make it more biodegradable by using a different type of polymer. So you can see how much more complicated. So this is tiny. So that is a very tiny needle. So this is about, I'd say, what's the size of this? Basically like your thumb size. So it's very tiny. But yeah. So you can see that we can do very complicated designs if we need to. So yeah, you kind of see, and obviously you will have either chips or something placed on here to make it, work. So let me just stop this video because I don't think we need to get the idea. I'll stop this and go back to sharing my screen. So let's go back. Okay, and obviously the next step is we're also looking at a lot of flexible and degradable polymers. So trying to see different types of polymers where we can remove the polymer itself and have reuse the metals. So basically collect them, have them drop to the bottom and recollect the metals and actually reuse them again so that we're not wasting pretty much anything. So we do have some from our Dr. Simon Holder's group, some polymers that are basically degradable with water. So if you have it as a wearable or for other different devices, that's an option, not just for medical technology, but for anything. Here, for example, I included one of our newest projects. This is a flexible wireless ECG. So this is fully wireless. And this is obviously, this one is not not a passive system for specifically, but it does use a printed battery as well. So everything is printed to order. So literally, you don't need to waste anything. You just make it for the patient as you need it. You don't have to manufacture, a bunch of them that are going to sit somewhere. You can literally print per order. So it's a very, this is pretty much where we're going when it comes to these making medical technology more green. Because the biggest issue with medical technology is that once there's patient samples, you can't just dispose of it. It has to be properly disposed. So going fully green is a bit more difficult when it comes to medical technology. Wearables, like something that's just on the skin, is a bit easier because there's not as much worry about having blood interactions or patient samples. It's a bit easier to work with when it comes to that. But actual stuff that's embedded, which is a lot of stuff that I do, going fully green is very difficult. And another project that I'm working on is I'm working with a big company on actual wound care management, and this will be a passive system that is multi-layer. So it's actually multiple sensors basically working together passively. So this is kind of where the future is going, and this is how at least I'm approaching going a bit more green, is making stuff that is per order, per request basically, rather than going towards full manufacturing everything. And so I want to just say, obviously, thank you to Robert Horn, who was part of the group and involved with this project. Dr. Campbell Goulet, who is part of the Ken Funkel group, who I worked with on the original project for the voice prosthesis, whose student also did all the antimicrobial research, and obviously Professor John Basher, who's the head of the tennis group. Are there any questions? I know there's a lot of information, hopefully I didn't make it too complicated.
So firstly, thank you very much, Viktorija. That was really interesting. We have had some questions come in, which I'll ask you, but I'll also, if anything comes up for the discussions, please chuck them in the chat on the Q&A and I'll pick them up and put it into there. Fascinating work. Thank you. Really interesting, diverse, and actually I think brings together some of the other stories that we've had throughout the week, like Dr. Maria Alfredson spoke yesterday about printing flexible batteries and things like that. Yeah. John Dickinson at the start of the week was talking about some of his wearable things that he wants in sports and things like that. So quite a nice story and bringing these things from the engineering point of view back together. Yeah. We've had a few questions, but I'll start off with the first one, which was around, you talked about these biofilms and these biofilms forming in various ways. Where do they form? Do they form on everything inside the body? The question is, are they forming in everywhere inside the body? Does it only form on these foreign bodies, like kind of implants? Is it only things it's supposed to air? Where do they form? So normally it's because a lot of it, so for example, Candida albicans, it's normally found in your throat. It usually doesn't cause any issues, but the moment you introduce some kind of polymer, it gives it a free-for-all, almost becomes pathogenic. So normally it's not pathogenic. You have it unless you have some kind of antimicrobial. So you start taking something an antibiotic, and it kills all the bacteria. Then the Candida albicans in your mouth is how often you get thrush in your throat. It kills off all the bacteria, so the Candida now has free space to roam, and it starts multiplying and growing out of proportion. So
So technically you have to introduce a device in order to basically have colonization of the device. A lot of the stuff that we talk about and we're worried for when it comes to prosthetics is literally your own body, like what's normally found on your body. That's what usually leads to infection of the devices because it's already there, but obviously the voice prosthesis is not supposed to be there. So the Canada just kind of takes advantage of that and starts growing. And it leads to basically colonization of the device and its failure, and sometimes also colonization and necrosis of the surrounding tissue. That is more rare though. Usually it's just a device failing.
So would you get it, for example, on a dental implant in the same way?
Yes, that's one of the reasons why there's a lot of research on trying to find dental work that is implanted with like silver, a bit of silver particles or different drugs and stuff in the dental, fake teeth. Basically, it doesn't work as well because it's only temporary. It does reduce, but only temporary. So it's kind of
that's because silver is antimicrobial, right?
Yeah, exactly. So that's why when we're talking about our siloxane polymers that we're developing, the whole polymer acts as the anti-fouling instrument. So it's not a drug that's implanted inside. It's the polymer itself.
I see that.
Yeah.
Very interesting. Another question, a couple of related questions here that I'll probably bulk into one a little bit.
Yeah.
How did you mentioned that there was a failure of prosthesis? Sorry.
Yes.
Try and pronounce that word correctly. Is that how were you establishing that? Was this something the patient was doing? Was it something they were saying, or how did you know?
So this is up to a speech therapist. Obviously, I don't work with the patients myself. Normally, they start having issues breathing and talking, so it's very difficult to close up the hole, and they're not being able to produce the sounds they're supposed to produce, and their breathing is affected because the little phalange, the little like valve thing that is in there, no longer can go up and down because it's being crusted over by the biofilm. So usually it's a voice it's a physician or a speech therapist who detects that there is an issue. The patient usually complains about, oh, I can't speak, I'm having issues producing sound, or I can't, breathe as easily or something along those lines. That's why it's very difficult to actually detect the colonization till that failure officially occurs. That's why this type of device, where it detects early when an attachment occurs to the surface, is very key. It's very good for detecting infection, basically.
I suppose related to this, and I suspect you when you get to patient trials, you'll probably find the answer to this truly. But you said there was this actually surprising differential between the failure time, like 2 weeks up to a couple of months or whatever.
Some people.
Do you know if there's a reason for this, or is it just how often they're reporting?
So there is some research, like I said, where they try to find out why some people have failure more often than others. So there's some studies in using probiotics to prevent this attachment and having colonization. Because if you have a bunch of bacteria and a bunch of lactobacilli and Candida together, the Candida can overpopulate it basically. So there is, I think they usually tell you to take probiotics, like yogurts, eat some dairy, avoid alcohol, because alcohol, like I told you, causes swelling of polymers over time. So it can actually cause what's called roughness to develop on the surface. And the moment you have surface roughness, the more colonization you can have. So there's some studies looking at different, there's not one cause for why you can have colonization on the device. So there's research still going on, I'd say, into this field. Like I said, I am not an expert on the bio side.
Yeah, sure.
That's where if you want to speak to somebody, go Campbell Gourlay. Dr. Campbell Gourlay this is his expertise.
Still very interesting. It's clearly stimulated some thoughts in the audience.
Yeah.
Let's go over to the sensor side. So we have a couple of questions here around the sensors.
Yeah.
How sensitive are the sensors, and could you, for example, detect different infections with a sensor? So obviously you've modeled this on one type of infection, but would in the future, would you be able to measure different infections? Yes,
that's actually where we're going next as well. So I'm really interested. So the newest project that I just finished is going into a prototype for a company with Spence Medical, is actually using tracheostomy tubing for Staph aureus. So the sensor was slightly adjusted to work with Staph aureus. The issue with some bacterial infections, they don't become as thick as Canada over time. So it's a bit harder to detect once they, because they're so thin. The thickness doesn't build up as easily. So it can be a bit harder to detect any other bacteria, whereas as Canada has a tendency, because they're much bigger, to be a bit easier to detect. So all of that will depend on how you make your. I'm trying to think of a way to say this without making it overcomplicated. So if you have the top of the surface and you have your sensor, so the way my sensor works, it uses almost like fringing fields, and the capacitance changes almost between like fingers. So there you can detect growth between these different layers of copper. Well, for now, the sensor is copper. It will be in real life will be silver, but right now we can actually detect the growth between, and because there's a change in the capacitance. So obviously changing the distance between these fringing basically fingers almost allows it to be able to detect it. So obviously it's not officially like a normal fringing field because it's a circular design and there's multiple circular interbits. But that's basically the basic function. It's almost like a fringing field capacitor, if somebody knows, if you're more technically minded. That's what the basic idea of it is.
So it's the same. I'll see if I can summarize it to my non-expert brain. If you have your two things, if whatever you change between that gap, we're measuring the change in whatever's happening between that, and that will change the resistance, whatever.
Yeah. Yeah, you can make it basically, you can change the capacitance of the system, and you can make it more prone to detecting something smaller or bigger, and because that will adjust the fringing fields around those two devices, basically two lines.
Fantastic. So I don't know which way around to ask these last couple of questions. If anyone else has any more questions, please do stick them in the chat. We have a couple more minutes, but let's go for this one. So you mentioned this, and we had this question yesterday, actually, with Dr. Alfredson, and I wonder if it's the same. But what's your vision for these? You showed your printing stuff at the end. I mean, obviously, I guess you're hoping to have these like printable in a clinic or something to deliver. But do you think there'll ever be a time when it might be even a patient prints these in their own homes?
Maybe the really long future, but for right now, so there's another company that we're working with. They have basically a separate component of their large. So Smith & Nephew has like a separate, unique component of their company where they do. You could basically, a physician could call them, say, I need this device. Can you make it for this specific case? So they do already have that available. So it could be basically built per order. I don't know about building it in a house and somebody's non-experienced, because the issue with these is that if something misprints, if you're not an expert, you might not know there's a misprint. So you put it in and then it's kind of a waste again. So I'd rather have a specialist lab that makes these, that is part of a company that make the devices, and they can print it for you. So that's where our actual wound care management is going to. It's going to be a multi-layer basically system where I can do moisture, temperature, and some other things to detect any kind of infection at a wound directly.
Fantastic.
So that is the future where we're going. So it will be all basically printed as well.
That's exciting. And I guess you've probably painted some of these images, but I guess a nice last question to wrap all of this up is, I mean, you've already shown how the technology has gone down, and you said secret, so we're not allowed to know yet, but the smaller than the 10 pence piece, and all of those things. But where do you imagine this field will be, or kind of you're sitting at the interface between technology and healthcare in like 20 years time, in 30 years time? Do you have an image of where it will be at all, or what you think? Will this be everything, every implant, everything will have sensors everywhere responding, or what do you think? Oh, I don't know if I've lost you. Look. Are you there?
Can you hear me? Yeah.
Yes. Sorry. Yeah, you're back.
I think it's maybe my internet, or I don't know. So yeah, the future that I'm seeing is that I am currently in talks with a lot of different companies to take their regular prosthetic devices. A lot of them are polymer devices. Like I said, I'm working on a tracheostomy tubing as well. I'm working on basically bandages for wound care management. So I really like, my personal goal is to take a lot of this, I'll call it dumb technology, like, just basic, you can't detect, it's up to the clinician. Basically, you take more, it costs more money for a clinician to watch this, for the patient having to go back and forth. There's also issues with compliance, not just at the hospital, patient not taking care of the wound, not taking care of the prosthetic device, but also compliance at home when the patient can't be watched to see, are they cleaning the prosthetic device? Are they changing? Are they actively not disrupting the wound? Are they, doing the exercises they're supposed to be doing to make sure the wound doesn't, have extra moisture or whatever? So my goal is to make a lot of the stuff smart, so that it actually can able to be detected to lead to better patient outcomes. So I'm already going there. Like I said, we have applications for different grants to work on all of this.
Fantastic. Well, that's very exciting, and I think a good place to leave it. So thank you very, very much for sharing that this evening. Thank you very much to the audience for asking those great questions and involving in this. And thank you for those of you who've been with us throughout the week as well. It's been an enjoyable week of lectures, and I think this was a brilliant way to wrap up. So thank you very much again, and thank you much to everyone, and have a wonderful weekend, everyone, and see you soon. Cheers.
Bye.
Thank you. Bye.